Hi, my name is Caitlin.
And my name is Casey.
And we did our project on corticosterone and how it predicts foraging behavior and parental care in macaroni penguins. The original research was done by Crossan et al. The macaroni penguin is our animal of study, and a macaroni penguin gets their name from their sassy yellow crest on the top of their head, as seen in the figure below. They have the black and white tuxedo look that is typical of most penguins. What makes them distinct is their large orange beak and red beady eyes. The macaroni penguin is the most common breeding penguin found. They live in rocky waterbound areas near the subantarctic and Antarctic peninsula. They are rarely found on land during their breeding seasons. The macaroni penguin feeds mostly on krill, but can sometimes eat small squid and small fish. Breeding of the macaroni penguin happens in late October and occurs in huge colonies. Hatching of the macaroni penguin eggs happens 33 to 37 days after fertilization. The chicks then go to sea when they are around 60 to 70 days old. What is corticosterone? Corticosterone is a primary glucocorticoid produced in the hypothalamic pituitary adrenal axis region of the brain. In birds, and in our case in penguins, corticosterone is the hormone responsible for behavioral and physiological responses. Corticosterone works by facilitating energy and the acquisition of nutrients, which allows behaviors and responses to stressors. A good real-life example of corticosterone at work in macaroni penguins is the positive correlation between parental care and their foraging efforts in the parents to provide for the offspring. In most cases, it is positively correlated. However, it can be negatively correlated when the production of corticosterone is too high. This leads to abandonment issues of the parents for their offspring.
Reproduction and parental care in macaroni penguins. As stated previously, macaroni penguins begin to breed in late October. They gather in large colonies and construct their nests in mud or in gravel. Their eggs are laid in November and are guarded by both parents until they hatch. The females then leave to forage while the males are left behind and guard the chicks. Corticosterone plays an important role in reproduction and parental care in macaroni penguins, as corticosterone influences the size of the chicks that are produced, where females with higher corticosterone levels produce larger, heavier chicks than those who have lower levels. This goes hand in hand with the corticosterone adaptation hypothesis, which predicts that those who can effectively upregulate endogenous corticosterone have higher fitness than those who cannot. As mentioned before, high levels of corticosterone can have a negative effect on parental care, where higher levels can increase the chance of offspring being abandoned by the parents. This goes hand in hand with the corticosterone fitness hypothesis, where those with higher corticosterone levels will have lower fitness.
This video shows the mass amount of colonies of macaroni penguins and how densely they live.
Foraging patterns in macaroni penguins. Female macaroni penguins are solely responsible for foraging and feeding their offspring, while male penguins are left behind in the nest and guard and sustain themselves via physical energy reserves. Females typically spend less time at the nest and are not predominantly responsible for guarding or brooding the chicks, just for foraging. Hypothesis and predictions. Based on the corticosterone adaptation hypothesis, as mentioned before, is individuals that can effectively upregulate endogenous corticosterone will have higher fitness than those who cannot, compared to the sham control, which is penguins that were implanted with an implant that does not release any corticosterone. Crossan et al. Predicted that penguins with the corticosterone implants will increase their foraging efforts, which means they'll spend more time at sea, they'll spend more time diving, and they'll increase the total number of dives they initiate. They'll also increase the total number of prey catching attempts they make. Implanted penguins would also have a greater gain in body weight at the end of their foraging trip and thus would produce larger chicks towards the end of rearing. They would also show an inverse relationship between corticosterone and prolactin. Females implanted with the corticosterone implants would show the lowest levels of prolactin.
To test this hypothesis, Crossan and al. Did a couple different experiments. The first experiment they performed was the field sampling protocol, where they sent a group of researchers to a Bird Island, which is an island mainly inhabited by different species of penguins. However, their focus for this research was the Macaroni penguins. The researchers followed a 500-member breeding colony of Macaroni penguins and set out to discover their departure and returning patterns in foraging. The penguins are not manipulated in any way, and they were simply just observed. In the second research, they did a blood sampling and corticosterone implantation. This is where female penguins were captured and blood was taken immediately at dawn on their journey away from the nests. So the blood was taken when they were departing from their offspring. Females were then implanted with a corticosteroid or sham implant. Again, the sham implant is placebo and contains no corticosterone. The purpose of the corticosterone implants were to temporarily increase plasma corticosterone levels above normal to then be monitored on a physiological level, and they wanted to monitor physiological responses like reproduction, parental care, and foraging. The third and final experiment that they performed was the hormonal assay and statistical methods. It was determined that prolactin plays a positive role in minimizing the negative effects of overproduction of corticosterone. In regular settings, the amount of prolactin was measured in each penguin, and the outcome from their foraging behavior was either determined as positive or negative based upon the amount of prolactin produced and the penguin's ability to forage and reproduce. From the research, they found some results. In each image, the gray dots represent the corticosterone implanted penguins, and the white dots represent the sham implanted penguins. So penguins with higher levels of corticosterone spent more time away from the colony for foraging. This is shown in figure A. The longer they stayed away from the colony, the more diving attempts those penguins made, which is shown in figure B. The more diving attempts they made meant they made more prey capturing attempts, which is shown in figure C. The more prey they captured means they gained more weight. The more weight they gained means they had more food to provide for their offspring, which resulted in heavier chicks, which is shown in figure D.
Some more results that they found is that the corticosterone implanted penguins had higher prolactin levels prior to leaving for foraging than when they returned from their trip, and that they also had higher corticosterone after returning from their foraging trip. So in figure A, it shows that the corticosterone implanted penguins had lower corticosterone before they left for their trip, and when they returned, they were measured to have higher corticosterone levels. And in figure B, they were shown to have higher prolactin levels prior to leaving for their trip, and they had lower levels after they returned.
That is all. Thanks.